Hello students, welcome to EPG Pathshala. I am Professor Pankaj Madan from Faculty of Management Studies, Gurukul Kangdi Vishwavidyalaya in Haridwar. Today we are going to talk on one module, that is Analysis of Variance and Experimental Design. An introduction to experimental, randomized and block design under the paper Quantitative Techniques for Management Decisions. After the completion of this module, the student will be able to understand the experimental design, the randomized design, the randomization of treatments, merits and demerits of the designs, application of the complete randomization and randomized block design, and what are the advantages of randomized block design. Experimental design is a way to carefully plan experiment in advance so that your results are both objective and valid. Ideally, your experimental design should describe how participants are allocated to experimental groups. A common method is completely randomized design, where participants are assigned to groups at random. A second method is randomized block design, where participants are divided into homogeneous blocks before being randomized assigned to groups. Minimize or eliminating confounding variables, which can offer alternative explanation for the experimental design results, allow you to make inference about the relationship between independent variable and dependent variable. Then, to reduce the variability and to make it easier for you to find the differences in treatment outcomes. The choice of experimental design depends upon the number and the nature of treatments under study. It also depends on the object of the experimenter. Another consideration for the choice of the design is the question of available resources. Let's give some consideration under which different designs are appropriate. Means what kind of this design is used in which situation. So let's talk about the first design, that is the complete randomized design. This design is appropriate when experimental material is limited and homogeneous, such as the soil in the pot experiment. Or you can say that it's like students in a class with almost all the students with above average level marks, maybe more than 60%. So if an institution is having a criteria that they will admit a student in a class with minimum 60% marks, then we say that we can use this complete randomized design. Then second is randomized block design. This is an appropriate when the fertility gradient of the field is in one direction. It can be adopted up to 20 treatments without appreciable loss of efficiency. Or in the other sense, you can say that this randomized block design can be used on the treatments in which you want to see the impact of different types of diets on the persons, but those persons are of the same homogeneous weights, or you can say the blocks of weights. Maybe you want to see the impact of different types of food on a category of patients or persons, like with a weight from 60 to 70 kgs, that may be a block 1, then 70 to 80 kgs block 2, and like that maybe 50 to 60 kg block 3. So in that case you use randomized block design. Third is Latin square design. This is appropriate when fertility gradient of the field is in two directions instead of one. It may be adopted for a number of treatments ranging from 5 to 8 or from 5 to 12 at the most. Factorial scheme. There are several factors with different levels to be studied simultaneously with the same precision. The factorial scheme may be adopted. Third is the split plot design. When the factors are such that some of them require large plots like irrigation, depth of plowing, sowing dates, etc., and may be suited with different precisions, split plot design may be used. Then last is incomplete block design. This can be used when the number of treatment is sufficiently large. In complete randomized design, this is the simplest way of design in which the whole experiment material is divided into number of experimental units depending upon the number of treatments and the number of replications for each. After that, treatments are allocated to the units entirely by chance. In case of field experiments, the whole field is divided into the required number of equal plots and the treatments are randomized in those plots. If there are 5 treatments A, B, C, D, E and 4 replications to each, the number of plots will be 20 and each treatment will be allotted to 4 plots selected at random by means of random numbers. Randomization of treatments. Here, since the number of units is 20, a 2-digit random number table will be consulted and a series of 20 random numbers will be taken, excluding those which are greater than 20. Suppose the random numbers 4, 18, 2, 14, 3, 7, 13 and like that up to 19. After this, the plots will be serially numbered and the treatment A will be allotted to the plots bearing the serial number 4, 18, 2, 14. Treatment B will be allotted to the plots bearing serial number 3, 7, 13 and 1 and so on for the other treatments C, D and E. In a similar way, the randomization can be done for any number of treatments. As far as structure of analysis is concerned, if we suppose that the treatments to be n and the number of replications to be r for every treatment, then the total number of experiments units will be nr and we can denote it with a capital N sign. In the treatments having varying number of treatments R1, R2, R3 up to Rn, the total number of units is equal to n will be given by n is equal to R1 plus R2 plus R3 up to Rn. In this design, the total number of degrees of freedom will be divided into 2 parts representing the independent comparisons. The two will be 2 independent sources of variation. One between treatments and another is within treatments. Within treatment components provide a basis for the estimation of error. Thus, the structure of analysis of variance will be as follows. Sources of variation, and in those sources of variation we have 2 categories, between treatments and within treatments, or sometimes we call it as between columns and within columns, and it is represented by n-1 and a bigger n minus small n, and we can see the F ratio upon in which we have calculated previously the F value of between column variance and within column variance. Here it is represented as Vt and Ve, where E represents the error. Here, since the total number of observation in n, the total degrees of freedom will be n minus 1. Similarly, treatment degrees of freedom will be n minus 1 one less than the number of treatments, and the remaining n minus degrees of freedom will be for within treatments or error. In this case, the standard error of the difference between the two treatment means based on R1 and R2, replication is estimated by the following relation, and it is representative by standard error where V into 1 upon R plus 1 by 2 and a root of that, where V is the pooled error variance or a error mean square. If R1 is equal to R2 is equal to R, the formula reduces to under root 2 VE upon R. The degrees of freedom for test T are the error degrees of freedom. If we talk about the merits and demerits of complete randomized design, then we can say that in this design, any number of treatments and replicates can be used. The number of replicates can also vary from treatment to treatment. Then the statistical analysis of data is very easy, as it remains easy even if the number of replicates are not the same for all the treatments. In this type of design, the method of analysis remains simple when the results from some units are missing or even rejected. The relative loss of information due to missing data is smaller than that of any other design. This design is especially useful in small experiments where the supply of experimental material is scarce and homogeneous, as the whole of the material is utilized in the experiment. The designer provides maximum number of degrees of freedom for the estimation of error as compared with other designs in this design for a given number of treatments and a given number of experimental units. But it has certain demerits also. There is one, and the main objection in this design, and that is on the ground of accuracy. Since there is no restriction on the randomization of the treatments, we cannot be sure about the fact that the units receiving one treatment are similar to those receiving the other treatment, and therefore the whole of the variation among the units enter into the experimental error. What is the application of complete randomization? Where it is used? This design is especially advantageous and appropriate under the following circumstances. First, where the experimental material is limited in quantity and homogeneous. Second, where it is expected that some of the units will be destroyed or will fail to respond. Third, in small experiments where the increased accuracy from the alternative design is not sufficient to exceed in importance the loss of error degrees of freedom. Or we can conclude that a complete randomized design is useful when you have a homogeneous sample, but the sample is large enough to be taken, in which even if few units are not showing proper results or destroyed, our results are not affected. If we talk about the randomized block designs, in the design, the whole experimental material is divided into homogeneous group, each of which constitute a single replication. And as I have told you previously, in case of we are seeing the effect of different diets on different age group of people, and those age groups are the blocks. Maybe one age group from 50 to 60 is one block, 60 to 70 another block, 70 to 80 another block. So each of these group is further divided into number of experimental units which are equal in all respects. So what it means is that this block contains a homogeneous kind of data. The treatments are applied to these units at any random process. In case of field experiments, if it is observed that the fertility gradient of the field is in one direction, the whole field may be divided into number of equal plots. The number of plots in each block is equal to the number of treatments, so that each block is replicate of each treatment. The following important points are to kept in mind for this design. In this design, the number of blocks must be equal to the number of replication fixed for each treatment. Second, the number of plots in each block should be equal to the number of treatments. Third, an important and essential point on which the attention is kept is that the experimental errors within each block are to kept as small as possible and the variation from block to block as great as possible. And that is why we can see the impact of different diets on different age groups, or you can say different treatments on different plots. In this way, all the treatments which are assigned to one block experience the same type of environmental effects and are therefore comparable. Randomization of treatments in each block should be fresh. Except the field experiment, in other types of experiments, the replicates can be identified with the sources of variations corresponding with position, time, classification of the experimental units. For instance, if experimental material is cows, they can be divided into groups according to breed, age, weight, location, number, etc. If in a herd of other sources of variation, except the lactation number are constant, the cows can be grouped according to the lactation number. One group will consist of cows. One group of cows. One group will consist of cows of one lactation number, and the other group will consist of cows of the second lactation number, and so on. Here the lactation number will be the replicates and cow will be the experimental unit. When we talk about the randomization of treatments, the treatments assigned to the units or the plots within each group, and that group is a block entirely at the random with the help of random numbers. It is important to note that for every group of randomization, it is important that randomization is fresh and same set of random numbers should not be used for all the groups. Different set of random numbers are to be used for different groups. Structure of analysis of variance. Taking the case of agriculture experiment, if we suppose that the number of treatments to be n and the number of replications to be r, the total number of degrees of freedom will be divided into three parts representing the independent comparison. And what are that? Between block comparison, between treat comparison, and random variation, which provides a basis for the estimation of error. And here you can see that the sources of variation are between block variation and the between treatment variation, and formulas are almost the same what we saw for the analysis of variance, only there is a difference in the degrees of freedom. Here the degrees of freedom is calculated by n minus r and n minus 1 and 2 r minus 1, and the total number of observation is nr. The total degrees of freedom will be nr minus 1. Similarly, since the blocks and the treatments are respectively r and n in number, their corresponding degrees of freedom will be r minus 1 and n minus 1. Let's see its standard error and critical differences. In this case, the standard error of the difference between the treatment means based on r applications is estimated by the relation SE, and that is under root 2 VE upon r. where V is the pooled error mean and the critical difference at 5% level of significance will be SE into T, which is the 5% for error of degrees of freedom. If some treatments receive extra replication, the general formula for the standard error is SE, and it is the under root VE in bracket 1 by R1 plus 1 by R2, where R1 and R2 are the number of replications of the treatments to be compared. Randomized block design has lot of advantages also. When the material is heterogeneous, the residual variance can be reduced by choosing blocks or plots such that the plots within any block are fairly similar. The design allows of another number of treatments and any number of replication, but when the number of treatments is very large, approximately 20 or more, the efficiency of error control decreases. Although a reduction in the number of replications lead to a larger standard error, yet it furnishes a result of small value at least. By means of grouping, more accurate results are usually obtained than with the completely randomized design. If we find that the experimental error variance is larger for some treatments than for others, we can obtain an unbiased error for testing any specific combination of the treatment means. Let's see one practical example and we can analyze the data. In the following table gives the yields in pounds per plot for 5 varieties of wheat after being applied to each of 4 plots completely randomized. So here you can see we have yields of different varieties of crop in different plots, and the analysis of variance. First we have found out the correction factor, and then we have found out the variations, the degree of freedom, and while calculating the F, which comes out to be 11.18 at 5% level of significance, and at 0.1 it is 8.25. Here F test indicates that there is a difference between the treatment means. Since the observed value of the variance ratio is highly significant at 0.1% level of significance. Now we wish to know as to which variety is the best and also which varieties shows the significant difference among themselves. This can be done with the help of critical difference and the confidence interval. Now looking at this table, F value, and if it is a computerized value, then you have a significance alpha value, and if the alpha value is less than 0.05, then we say yes, there is a significant difference in the yields of different varieties. For manual calculation, you can see the standard error of the difference between two different treatment means is 1.317, and the critical difference is 1.317 into T at 5% levels for degree freedom, that is 2.131. So it comes out to be 2.81. Here F test is the analysis of variance, indicates significant difference between the varieties, and therefore we are justified in comparing the individual varieties with help of critical difference. You can see in another agriculture experiment that a yield of 6 varieties, and this is a question actually of block design in which you see 6 varieties of crops along with the plan of the experiment, and the number of blocks is 5. Plot size 1 by 20 acre, and the varieties has been represented by A, B, C, D and F. So you can see different varieties divided in different blocks. So here we are also interested to know the impact of blocks as well as varieties. So we have divided the crops and their output according to different plot sizes as well as varieties. And you can see here the table where correction factor is 2154.7.2 and variation due to blocks is 61.5 and due to varieties it is 10167.2. Error we have found out, the manual way of calculating, and here we have a table with different significance values. So 97, it is clear from the table that this high observed value of F is significant at 0.1% level of significance, which proves that there are significant difference between the variety means. We have to test the significance of the difference between the individual varieties, and that will be done with the help of critical differences. And here on the right hand side of the table you can also see the F table values which has been shown at different degrees of freedom or different levels of significance. So the students, let's now summarize what we have learned in this module. Randomization, replication and error control are the three main principles of design of experiments. Randomization, which defines the manner of allocation of sample or population or treatments to the experimental units. Replication, which specifies the number of units to be provided for each of the treatment or population. And error control is one which increases the precision by choosing appropriate type of experimental units and also their grouping. We have seen that in a completely randomized design, no local control is adopted except that the experimental units should be homogeneous. Usually, when the experiments required a large number of experimental units, completely randomized designs cannot ensure precision of the estimates of the sample or the population. An improvement of completely randomized designs can be obtained by providing an error control measures as a randomized block design. The error control measures in this design consist of making the units of each of these groups, and these groups should be homogeneous. These groups are commonly known as blocks. Block A, Block B, Block C and like that. And the experimental units in these blocks are known as plots. This type of homogeneous grouping of the experimental units and the random allocation of sample separately in each block are the two main character features of randomized block design. I may like to again specify that the treatments are the three different, when we call it as three different populations, they are the three different treatments or the three different samples. Thank you.